Okay. Thank you, Professor Yong, for your kind introduction. I also would like to thank the organizing committee and SAL for inviting me to be here. And also congratulations for all the excellent work you have done in the past 40 years, and many more, in the future. So I would like to share with you 3 clinical trials we have done in the past 10 years also. And so we use a high-fiber diet to change the gut microbiome of patients with obesity, type 2 diabetes, and then try to understand the role of gut microbiota in human metabolic health. However, I would like to present these clinical trials from the perspective of microbiome data analysis, and so which we call it reference-free and relationship-focused strategy for analyzing the microbiome data. And talking about microbiome data analysis, Dr. Robert Knight has laid the foundation of microbiome analysis for the whole field. He's the pioneer for this field, and we have been using all kinds of tools he has been developed, along even now, and at almost like a daily basis. And looks like this. I have a conflict of interest to declare. I'm co-founder of Nutritional biotechnologies company. And we know gut microbiome is very complex. It's just like the Amazon jungle. And such a system can be considered the so-called complex adaptive system. Such a system, they have many, many components. However, these components are not randomly mixed together. They have, different components may connect together to form a higher level structure called modules. And then different modules will connect each other to form the network. And then the whole network is responding to the environmental changes in a concerted way. So this is a so-called complex adaptive system. And then if you come down to gut microbiome as a complex adaptive system, the basic components would be microbial cells. Then different types of microbial cells, they work together to form a higher level structure. In ecology, we call it guilds. So each guild is a coherent functional group of bacteria. They come from all different taxonomic background, but they work together as a coherent group. They increase or decrease together and showing co-abundance behavior. And so the best way to profile microbial cells is to get their genomes. So just mentioned by Dr. Robert Knight briefly in his talk, if you can do deep metagenomic sequencing, you can assemble, de novo assemble genomes of microbes in your sample, which is called metagenome assembled genomes. So we do this in a way that we have 1% ANI difference between genomes as the resolution. So for each unique MAG, we actually give a unique identifier, universal identifier. So in this way, we don't need any database. So this is what we call reference-free, as he mentioned also in his talk. So this also gives you a close to string level resolution, because we know microbial functions are actually string specific. So we need to increase the resolution of our analysis. After we assemble high quality MAGs, high quality genomes, then we need to look at the ecological behavior of these genomes and try to understand who works with whom as a guild or as a module. And then try to understand the module-to-module or guild-to-guild interaction as a network. So this is the overall framework that we try to use to analyze our high-fiber dietary interventional data. And the way we do high-fiber diet intervention is because if you can do experimental intervention to change the microbiome and then see if there are any associated changes of host health parameters or disease parameters. This gives you a very high level of causal evidence and to understand the role of gut microbiota in human diseases. So the particular high-fiber diet we use consists of whole grains, traditional Chinese medicinal food, and prebiotics. So we provide a balanced nutrition to human, but at the same time, we included a very high amount of very diverse fiber into this diet. So the idea is to use the high-fiber diet to change the microbiome so that if we can see if we can improve the disease. So I myself, by the way, was the first volunteer. So I lost about 45 pounds and recovered from metabolic syndrome by testing this diet. And then we conducted a few clinical trials, most of them published. So this one was published in 2015. It's targeting two groups of morbidly obese children. One is have Prader-Willi syndrome. So this is a genetic disorder with morbid obesity as a major phenotype. And when we do the hospitalized nutritional intervention in Guangdong Women and Children's Hospital, we accidentally found out that 1/3 of the morbidly obese children turn out to be Prader-Willi syndrome patients. And they also responded very well, surprisingly, to this high-fiber diet. And for example, this boy stayed in the program for 285 days, and he reduced from 140 kilograms to 83.6 kilograms. And then when he continued the diet at home, after 430 days, he was 73 kilograms. So not only he lost about half of his initial body weight, but also recovered very well from all the major metabolic syndrome issues. So eventually we recruited 17 PWS children on the program for 3 months, and simple obesity children without a known genetic reason, 21, for 1 month. And for 3 months, they lost about 20% of initial body weight. For 1 month, they lost about 10% initial body weight and recovered from metabolic syndrome. And particularly, they showed alleviation of inflammation. By using these several different inflammatory markers, they all showed reduced inflammation. Just echoing the currently very hot topic of anti-inflammatory diet. But basically, this high-fiber diet showed anti-inflammatory effect. And the lipopolysaccharide binding protein in the serum is actually a surrogate marker to show the antigen load, particularly endotoxin load in the blood, and it's indeed reduced. And we transplanted the microbiota from baseline and 3 months after intervention from the same individual to germ-free mice. And we found that the baseline microbiota from the patient induced inflammation first, and then induced excessive fat accumulation in the recipient mice, while the after intervention microbiota didn't have such detrimental effect. So this is a piece of causality evidence to show that the before intervention microbiota had the capacity to aggravate or induce disease, while the after intervention microbiota didn't have such capacity. And so we metagenomically sequenced all the fecal samples, and we assembled high-quality genomes, and we selected 161 high-quality genomes because they show up in more than 20% of the samples, and altogether, they amount to more than 65% of the sequence we generated. So these 161 genomes represent 161 bacteria which are both predominant and also prevalent in the cohorts of our patients. And then we did a co-abundance analysis among these 161 genomes, and we were able to delineate them into 18 guilds. The same colored group is one guild. If they connected with red line, they co-occur. But if the two guilds connected with the blue line, they co-exclude from each other because the members in the same guild change in the same direction. So we can lump them together to get guild level abundance variation of these 18 guilds. And then we correlate the guild changes with the disease parameters. 3 guilds showed negative correlation with disease. 9 guilds showed a positive correlation with disease. And the rest of 6 guilds showed no correlation. So basically, this is the network mapping of the major members of the gut microbiota, not only with each other as 18 guilds, but also with the host. And the 3 blue arrowed guilds, they were increased by the high fiber diet, showed a negative correlation with disease parameters. They are potentially beneficial, protective to the host. While the 9 red arrowed guilds, they were reduced by the high fiber diet, and they were positively associated with disease parameters. So they were potentially pathogenic or detrimental. So this is the way that we analyze the microbiome data for this clinical trial. And up to this moment, we didn't query any database. We didn't need any database. Come to this point. After we get this guild, we identified the health relevant guild. Then we query the member of each guild to the database. So for some guild, we say that some members are well-known beneficial bacteria, such as bifidobacteria in this beneficial guild. But in the same guild, we have some less well-known bacteria as beneficial, but they are part of the beneficial guild with well-known beneficial bacteria. So we have good reason to believe that they are also beneficial bacteria, even though they are not very well studied. And in this guild, it's potentially beneficial. However, some of the genome at that time, we even couldn't give a phylum level taxonomy. So that means they represent very novel, understudied bacteria. However, we can still identify them as part of a beneficial guild. So that means our approach didn't miss novel bacteria just because you cannot give a name, you throw them away in your data analysis. That doesn't make much sense. So this is also the first paper to show that there's actually major members of gut bacteria, they organize themselves as guilds and not as taxa. So that's why we should analyze our microbiome data based on ecological behavior, not based on their taxonomy. And the second study I would like to share with you, published in 2018 in Journal Science, it's a randomized controlled trial using the same high fiber diet, but in type 2 diabetes patients. Because this is a RCT trial, so we controlled all the major things and make high fiber diet. High fiber intake is the only difference between the two groups. And so after 3 months on the high fiber diet, the treatment group showed actual clinical benefit compared with the control group. Because of the RCT design, we can only attribute the actual clinical benefit to the actual intake of dietary fiber. And so we established a causal link between the treatment and the clinical outcome. We also transplanted the microbiota from the same patient before and after the intervention. And we showed that the before intervention microbiota induced type 2 diabetes-related symptoms in the recipient mice. Well, after intervention microbiota didn't, from the same patient, didn't induce disease-like symptoms. What does this mean? Because the recipient mice, they had no genetic defect, they were on normal diet, so basically they would not develop any disease symptoms. However, after receiving the microbiota, baseline microbiota from the patients, they started to develop disease-like symptoms. This can only show that the disease was induced by the dysbiotic gut microbiota from the patients. And so short-chain fatty acids are produced by fermenting dietary fiber by bacteria. So we also showed that butyric acid was significantly increased in the treatment group, while maintained at a low level in the control group. And the butyric acid can bind to the L cells receptor, GPR41-43 receptor, and induce GLP-1 production. GLP-1 can stimulate insulin production. So indeed, we see GLP-1 increased and insulin increased. So this is probably one mechanism why high-fiber diet can help type 2 diabetes patients. And we also see reduced pH or acidification of the gut. That's a dramatic change of the gut environment. So the gut microbiota must respond dramatically to the reduced pH of the gut. So we assembled 141 high-quality genomes from the metagenomic data, and they were present in more than 20% of the samples. They represent more than 60% of the total sequence we generated. So these 141 genomes, they represent the most prevalent and predominant members of the gut microbiota in the cohorts of patients we studied here. And more importantly, in each of these 141 genomes, you can see at least one pathway for producing short-chain fatty acids. So this means that they also have all kinds of genes for using carbohydrates. So this means that genetically, these 141 genomes have the capacity to use some kind of fiber and produce short-chain fatty acids and grow. However, when we provide the high-fiber diet with high amount of very diverse fiber to the patient's gut, surprisingly, only 15 promoted by the high-fiber diet, and 47 actually reduced by the high-fiber diet. The majority, 79, actually remained unchanged. So if you look at these 15 positive responders showed a negative correlation with the 47 negative responders. And when we look at the genomes, the functional side of the genomes of the negative responder and positive responder, we realize that the positive responders, they grow quicker because they produce more ATP than the negative responders from the same amount of carbohydrates, and they are also tolerate to low pH. When more short-chain fatty acids start to accumulate in the gut, pH reduced. And so that's probably why, and after some time, this group of bacteria become predominant in the gut, while this group actually reduced lower than baseline. And interestingly, the diversity of these 15 strains and the total amount they can reach, when we combine this into a short-chain fatty acid producing index, show the negative correlation with HbA1c. So the higher the diversity and the total abundance you can achieve after you take the high-fiber diet, the lower the HbA1c after 3 months you can achieve. So there's a negative correlation between these two parameters. And if you look at the mechanism, so basically the increase of the acetic and butyric acid producers, these 15 strain, change the gut environment in a way that reduce these 47 negative responders. Well, these bacteria, they produce endo hydrogen sulfide. These were inhibitors of GLP-1 production. And more importantly, they also produce endotoxin, and these are actually inflammation inducers, and inflammation can damage the insulin receptor. So because of this, when you reduce endo and hydrogen sulfide and endotoxin producers, you further release more GLP-1, you increase the insulin receptor sensitivity so that you get even better reduction of HbA1c and better glycemic control. So the last study I would like to share with you is actually in our previous clinical trial, 3-month clinical trial, we get our patients back. After 1 year, they stopped the high-fiber diet. And then we found that the disease, the microbiota, in the first 3 months moved from here to here, but after 1 year they stopped, the microbiota changed back, and the disease also came back. So we give a high-fiber diet for 3 months, and then we take it away. So this treatment induced dramatic changes of individual members of the gut microbiota. But we would like to see which two genomes can remain the stable correlation, no matter how dramatically you change the environment and change their abundance. So if two bacteria can stick together, either positively or negatively, they have a stable ecological relationship. No matter how dramatically you change the gut microbiota, that's an indication that these two bacteria must have co-evolved with each other and also with the host for millions of years. Because it's not random, it's not possible if two bacteria just live together for a few months and then they can form a very stable relationship. So based on this hypothesis, we assembled 1,845 genomes out of the data set, the 3 time points, and then we established, we selected 477 prevalent genomes, and we used these 477 genomes to establish co-abundance network for each of the time points. And then we look at genome pairs which keep the same ecological relationship across all the 3 time points. And interestingly, most of the possible genome pairs showed no correlation at all. More than 92% of the genome pairs showed no correlation at any of the time points. This is an indication that it's not easy for two bacteria in our gut to form an ecological relationship, even presently. However, a very small number of genome pairs, they showed either positive correlation at all the 3 time points or negative correlation at all the 3 time points. And very stable ecological relationship. And even though their abundance changed dramatically from time to time because of the high fiber intervention. And then we found that these genome pairs actually form 2 guilds. Inside each guild, it's positive correlation. Between the 2 guilds, it's a negative correlation. And also, we have only 141 genomes forming these 2 guilds out of nearly 2,000 genomes. And these 2 guilds, members of these 2 guilds, they are actually highly correlated with all the other bacteria. They are not just the most stably connected bacteria in the ecosystem, and they are also the most highly connected and the most widely connected. So they are the backbone of the ecosystem. If you remove these 141 genomes, the whole system just collapsed, even though you only remove less than 10% of the members. And these 2 guilds showed a negative correlation. High fiber diet increased guild 1 and decreased guild 2. Remove high fiber diet, guild 1 decreased, guild 2 came back. If you look at the genomes, guild 1 had more genes for degrading fiber and producing short-chain fatty acids, particularly butyrate, while guild 2 actually had many genes for antibiotic resistance and also many genes for virulence factors. So genetically, guild 1 potentially beneficial and guild 2 potentially detrimental. And then we use this 141 genome to show that this 141 genome can be used as reference genome to establish a machine learning regression model to predict 41 out of the 43 clinical parameters of the patients we studied. And then if we use this as a 141 genome as reference genome to examine other published case control studies, like the type 2 diabetes study, both in healthy cohort and the patient cohort, you see the existence of similar structure and genomes. You can also establish a machine learning model to predict the disease from control. And we also tested this 141 genome in other diseases, including atherosclerosis, liver cirrhosis, ankylosing spondylosis. It's the same thing. The bacteria, this key bacteria exists in all these published data, and just by using this group of bacteria, you can actually classify disease from control, and including Parkinson's disease, schizophrenia, colorectal cancer, IBD, and hypertension. All these case control study published by other people, by using our 141 reference genome, we can actually detect the signal and use this data to do classification of their samples. And so we suspect that this is a highly conserved microbiota among people, and also it's possible to do intervention by specifically providing nutrition to promote the foundation guild, and you may help alleviate various diseases. So now we have a product which can promote the beneficial guild, and in a few clinical trials. And if patients, some patients may have lost this structure, then you need to do transplantation to specifically reintroduce the beneficial guild, so that, and also give the nutrition. And so this is the way to show you that by using this genome-centric, reference-free, and guild-based approach, particularly looking for stable genome pairs, we may land on some very key members of the microbiota, which may show fundamental effect to human health. And thank you. Yeah.